My name is Kathy, and I think you know this already. And of course, a big shout out to give a big shout out to Liberty. You can see the blue background. They really care. You know that Liberty have been doing this for like 28 years, guys. 28 years. So that's 10 years longer than you've been alive. They've been caring about education. And you know what? One day when you guys are old and you are, well, not my age because I'm very old, but like when you're in your 30s, you know what? You're going to be having children and they're going to be assisted by Liberty as well. I mean, how awesome is that? So there you go. I mean, they really do care. And if they can take care of kids and education, believe me, they can take care of you as well. So there you go. We don't have our motivation-y thingy today. I sent it through very late, so I'm sorry. I'm hoping that they'll find it. But yeah, it's my fault, so please. But it was basically to say you guys must work hard, you must stay focused, and listen, if anybody offers you an exam paper that's leaked, don't look at it. Because no matter how strong the temptation is, do not look at it. And I'll tell you why. If you get caught in any way having cribbed in your matric final exam, I promise you now the penalties are not worth it. They're not worth it. Okay, so please don't. It's like if someone comes to you and offers you R1 million to do something bad, don't do it. Because the penalties are just too high. Just don't do it. It's morally not correct anyway. Believe in yourself. We've done a lot of work here with you, a lot, okay? And if you've listened to everything that we've worked through this year, I promise you now, guys and girls, you are going to rock those life science exams. Okay, we're focusing on paper 1 revision today. Okay, so I want to show you this. For paper 1 in this exam, you are going to, okay, so paper 1, all right, you're going to write for 2.5 hours. So you start at 9 and you're going to write until 11:30, okay? It's going to be 150 marks. And that 150 marks and that 2.5 hours is going to fly by. Now, this is how it's broken up. You're going to do meiosis, okay? And in meiosis, that's going to be approximately 11 marks. Okay, then you're going to do reproduction in vertebrates. Okay, and that's going to be about 6 marks. Now, what they normally do is for reproduction in vertebrates, you end up with short questions. So it'll be multi-choice, or it will be still in section A. All right, or part of question 1. And, you know, it'll be 1.3 or 1.4 or 1.5, and they'll ask you questions about, for example, the difference between ovoviviparous, viviparous, oviparous. They'll ask you about your amniotic egg, those kind of things. That's what they're going to ask you. All right? Okay, let's just see here. Then we've got human rep. And human reproduction, that's going to count 31 marks. Now, 31 marks is a lot, guys. It's 1/5 of your exam. So make sure that you know all of that stuff. Okay, then we've got responding to the environment. Okay, and that responding to the environment is your brain or your central nervous system. And your reflex action, and the eye, and the ear, and that makes up 40 marks of your paper. It's a lot. So your eye and your ear will probably be about 10 marks each if you look at all the shorts, etc. Your reflex action and your brain and the spinal cord, that makes up the other 20. Now, if we look at the endocrine system, and look at homeostasis. They list them as different sections, which they are, but they actually are combined too, and that is 15 and 11. So that gives you 26 marks. It's quite a lot of marks again. And then you've got responding to the environment plants. Okay, and one of the questions that I saw that Mbali loaded for today is about plants. It's phototropism and geotropism and auxins versus gibberellins, etc. So easy peasy. That's an easy 11 marks. And then you've got the last part is going to be human impact. And human impact you did in grade 11. And it's going to count 25 marks as well. And it's actually very easy. It's all about the basics. It's about water and about how we look after our area that we live in. Okay, so it's water, it's the environment, it's looking after our planet. Okay, so guys and girls, there is no reason for you to do badly. All right, let's look at our first question today. Okay, what have we got here? Says the diagram below represents the, let me get a different color here. Okay, the diagram below, and oh, by the way, this is Kesman's question. Okay, and start again. The diagram below represents the distribution of chromosome pair 21 as it appears in gametes at the end of meiosis 2 in a human male. Okay. Now, people, check this out. Remember, when we have meiosis, there are two parts. There's meiosis 1 and meiosis 2. During meiosis 1, we start off with a diploid cell. That diploid cell undergoes reduction division during meiosis 1, and we then end up with 2 haploid gametes. What happens, or 2 haploid daughter cells. Then it undergoes meiosis 2. And during meiosis 2, we end up with haploid gametes as well. Now, in the male, we're going to have, if this one carried the X chromosome and this one the Y chromosome, these two are going to result in daughters. And these two are going to result in the Y, which is males. I was going to put males, it's sons. So daughters and sons, so we'll have sons. Okay? Or a whole variety of combinations here. The important part here, though, is that this and this gamete, okay, they each are haploid. They'll have 23 chromosomes each. That's normal. That is perfect. Sometimes there's a mess. Sometimes there's an error. Now, if you think that meiosis occurs, I mean, males produce 90 to 150 million sperm a day. That's a lot of little spermies. So a lot of meiosis occurring in the seminiferous tubules of the testicles. But now there's a mistake. And that mistake is when chromosome number 21, because remember, there's chromosome pair number 1, 2, 22, and then you have your X and your Y chromosome in males. That makes up number 23, pair 20. This is the 23rd pair. Okay? So, when 21 appears in a gamete at the end of meiosis in a male, look at this. Well, here we have 2 chromosome 21s. Here we have 2 chromosome 21s. If that is the case, it means that those gametes are going to now not have 23 chromosomes, they have now 24 chromosomes in total instead of 23. Why? Because pair number 21 didn't separate. So I've got 2 times 21 there, and I've got 2 times 21 there instead of only 1. Okay, so this is a problem. And that is called non-disjunction. Non means didn't happen. Disjunction means to disengage, to separate. Disjunction. So if there was non-disjunction, it means they didn't separate. Okay, and it was pair number 21. That is called trisomy, or causes trisomy 21. Okay, when 21 doesn't separate, because if this little spermy or this little spermy joins with an egg cell or ovum that is normal at 23 chromosomes, you're going to end up with 24 plus 23 is going to be 47, and that is a big problem. Okay, so now, if two 21s going to this gamete and two 21s going to that gamete, well then C and D are minus a 21, and this one is minus a 21 chromosome, which means these little gametes are now going to only have 22 chromosomes. And that is a huge problem, because if those little sperm cells fertilize an ovum or an egg cell, they now sit, instead of 46, they're now going to have 44 chromosomes, and it will never ever materialize. The body will abort. The body will spontaneously abort that egg. I mean, that zygote. All right, so let's look at our questions. And it says, explain why the gametes represented in C and D do not have any chromosomes. Well, that's very easy, and we've already explained it, but let me write it out for you. So let's get a bit of blue screen here. Okay, so we want to know why, and that was question 2.1.1. And why would it be? Because due to non-disjunction, okay, the chromosome pair 21 did not separate during anaphase 1, okay, or the chromatids of chromosome 21 did not separate during anaphase 2. Okay, and if that is the case, you're going to end up with 2 chromosomes instead of only one, number 21. So as we saw in the diagram here, we've now got 2 21s there and 2 21s there, which gives a total of 24 chromosomes overall. And that is a mess, because when we add 24 from the sperm cell and 22 to the egg cell, well, what happens then? It's a problem. So when this happens, so when they don't separate and they don't separate, there are 2 number 21 daughter, oh, for heaven's sake, daughter chromatids in the resulting gametes. Okay, just remember this. We start off, as I showed you, with a diploid cell. That diploid cell is going to undergo meiosis 1. Okay, so you're going to have prophase, anaphase, prophase, metaphase, anaphase, and telophase. The end of telophase, you now end up with 2 haploid cells. Okay? During anaphase, you've got your your whole chromosomes are going to pull to the poles. Metaphase, you have a double row. at the equator. Prophase, you're going to have your crossing over. Okay? So you'll be able to see all of these and that it's meiosis 1. Now these guys here have to now undergo meiosis 2. And when they undergo meiosis 2, we end up with 4 unidentical, so there are 4 unidentical genetically different daughter cells. And they are haploid. So that when I take the male from the sperm cell gamete and the female from the ovum gamete, and they join together during fertilization, I'm going to end up with a zygote that is 2N. And the zygote will then grow and develop and undergo mitosis, and it will just grow and grow and grow. So we'll get 2 cells to 4 to 8 to 16, and then we'll end up with a blastocyst, and we'll have implantation. Okay, so that is the process. So if we go back here, that is why C and D, nothing, because you've got 2 21s sitting in each of these gametes. Okay, now, if gamete A is involved in fertilization, describe how this may result in Down syndrome. This is what you're going to have. You're going to have the sperm cell, which is haploid, contains 24 chromosomes, okay, 2 copies of chromosome number 21, okay. Your the ovum, or the egg cell, is also haploid, and it contains the normal 23 chromosomes. And I'm going to put in brackets here, 1 copy of chromosome number 21. Okay, so what happens now? You have fertilization takes place. So you have 24 chromosomes plus 23, which is normal from the ovum, okay? That's going to give you 47 chromosomes when it is diploid. So what do we have here? We therefore have 1 extra copy of number 21. And that is called, so hang on, so therefore we have 3 number 21. Therefore we have trisomy. Tri means 3 and somy is chromosomes. Trisomy 21, and that causes Down's syndrome. Okay. Do you understand, guys? I hope so. So you've got 44, and something else that I want to mention here. All the textbooks will tell you that females, as they get older, they stand a greater chance of producing ovum or ova egg cells that have 24 chromosomes. In other words, copies of or a non-disjunction of chromosome 21. Okay, as they get older. So the chances of having a Down syndrome child increases. But nowhere do they tell you that males can also do this. It can happen in the male as well. So your sperm cells can also contain 22 chromosomes or 24 chromosomes. It's not just the female that causes or produces because of a woman that the baby is a Down syndrome child, men have just as much of a, well, not just as much of a chance, they have a far lesser chance, but it does occur in males as well. And this question, when it did appear in the exams, threw a lot of learners because they thought, but the sperm cell can't happen in a sperm cell. Yes, it can. Okay. Any cell where you have trisomy of a chromosome, in other words, 3 copies when fertilization has occurred, is going to be trisomy. So it could be any of the chromosomes. All right. But if it is chromosome 21, it results in Down syndrome. All right, let's just check our other questions. All right, then it says, due to the process of crossing over, the chromosomes in diagrams A and B appear different from each other. Can you see? So there we've got a piece there and that, and there and that. Which why? Because they crossed over. So identify the phase of meiosis during which crossing over takes place. Well, that's going to be prophase 1. And people, something else to recognize, if they give you meiosis diagrams in an exam, you must be able to recognize them. Sharon actually asked for me to help her with the difference between meiosis and mitosis. Meiosis 1 and meiosis 2. That's one of the differences. In meiosis 1, prophase 1, you can actually see the crossing over. And in prophase 2, there is no crossing over, but you can see that the pieces have been swapped. All right, then describe the events of crossing over. I'll do that now. And explain the significance of crossing over for natural selection. Well, what does it do? It's going to cause, let me just do this question first. It causes variation because pieces of genetic material are swapped between the homologous chromosomes, and you can put specifically the non-sister chromatids, resulting in a different genetic makeup from the original cell. Okay, so that's easy peasy. Now, describe the process of crossing over. People, you've got to know this. Okay, watch. Okay, I'm going to draw. So we've got chromosome 1. There we go. This is a chromosome pair. These are homologous chromosomes. So what is a homologous chromosome? They're going to have the centromeres in the same position. They're going to be the same length. They're going to be in the same position. They carry the same genes. Okay. This one's from the mother, so it's the maternal chromosome. And this is the paternal because it's from the father. Okay? So what happens during crossing over? And watch. First thing, number 1, okay? 2 homologous chromosomes line up next to each other. Okay, so here they are. You see, they're next to each other, there and there. All right, so what happens now? We have these two. These are sister chromatids. Okay, and these two, this one and this one, are non-sister chromatids. Remember, sister chromatids belong to the same chromosome. Here you go, your sister chromatids. They have undergone replication, so these two are going to be identical. Okay, So what happens, these are non-sister, the one from the maternal and one from paternal. So they lie next to each other, and then they do this. Okay, so let's color this one in, so you're going to be able to see what happens. Okay, so that's the one maternal chromosome. Here's the paternal chromosome. So the two cross over. And then what happens? So they cross over at the chiasma. That is this point here where they cross over. The chiasma. And what crosses over? Sorry, I didn't. The non-sister chromatids cross over at the chiasma, and then your point 3, okay, is they now swap genetic material. And then point 4, when they separate, genetic material is in place and genetic variation can occur. Why? Because they're not the same as the parent ones were. Okay, so this now is going to look like this. We're going to have our chromosome. Okay, I'm going to draw it a bit smaller so I don't have to color so much. There. And we're going to have the other chromosome. So the paternal chromosome is going to look like this. Okay, and they're going to have a little shaded part there because this piece went to there and this piece went to there. They swapped genetic material. So they are no longer the same as the original set of chromosomes. Okay, and that happens during crossing over. All right, we're going to go for a quick ad break and I'll see you all in a minute.